In this mini tutorial we're going to think about the function of the cerebrospinal fluid. So this is the CSF which bathes the brain and the spinal cord, and it has a number of essential roles in the normal structure and function of the central nervous system. Probably its major role is in physically supporting the weight of the brain and the spinal cord. And the way that I imagine its role in this is by thinking about a jellyfish. So if you imagine a jellyfish in the sea, so here's a jellyfish here like this, it's got all its tentacles coming down, supported by the seawater. So it's floating in the seawater. Now you can imagine if that jellyfish gets washed up on the beach, what does it look like? Well, it looks like a fairly pathetic sight. It's all flattened down, and because it can't support its own weight in the absence of water. So water helps to support the weight of a jellyfish, and cerebrospinal fluid supports the weight of the brain and the spinal cord. And without that, the very soft consistency of the brain and the spinal cord would result in them essentially flopping down under the force of gravity. So one function of the CSF is to support the weight of the brain. It also has important nutritive functions. For example, the CSF has high levels of glucose, and this does help to nourish the cells on the surface of the brain and the spinal cord. And in addition, there are certain immunological roles associated with the CSF. It used to be thought that the central nervous system had virtually no immune cells within it, but now it is known that actually there is a constant turnover of immune cells in the central nervous system, and you can even find those in the cerebrospinal fluid. So those are the three main functions of CSF. Now let's go on to think in more detail about how CSF is made, how it circulates, and how it's reabsorbed. So if we're going to think about the production of cerebrospinal fluid and its circulation, let's draw a very, very simplified diagram of the brain and a bit of the spinal cord. So here I'm drawing the surface of the cerebral hemispheres. Here is the brain stem with the spinal cord there. As you should recall from Bio 273, the central nervous system is completely enveloped with three layers of meninges. However, the only layer of meninges which is relevant to our discussion of CSF in this case is the arachnoid, which I'm going to represent in green. So the arachnoid is relatively loosely applied to the entire surface of the central nervous system. So you can imagine that with the arachnoid it's a bit like if you were to put the brain in a plastic bag. It wouldn't go down into all of the sulci. It would just sit on the surface. And of course the brain enveloped in the meninges is in turn enveloped in the skull, which I'm not going to draw on in this case. So now let's draw on the ventricular system. Now the concept of the ventricular system is relatively straightforward to get your head around, particularly if you go back to your early lectures on development. And you should recall that the central nervous system, that is the brain and spinal cord, is derived from the outermost layer of the embryo, called the ectoderm. And what happens is that the ectoderm folds in on itself like this. Okay? So ectoderm folds in on itself, and you can see that it's forming a cylindrical structure here, so we're looking at a cross-section through it here. And ultimately, this cylindrical structure completely pinches off and separates from that surface ectoderm. And this structure here, which has been formed, is called the neural tube. Now, the neural tube becomes the brain and the spinal cord. And if you understand how the brain and the spinal cord develop, that is from a tube, then the obvious implication is that the brain and the spinal cord are hollow. Now when you're looking at brains and spinal cords, you don't automatically tend to realise that they are indeed hollow, but they are. So let's draw on what the cavities within the brain and the spinal cord look like. So here I'm drawing on parts of the ventricular system. Okay, and what I've just drawn on there are known as the lateral ventricles. So those are the cavities found within the cerebral hemispheres. They're called the lateral ventricles, and they're filled with cerebrospinal fluid. The lateral ventricles are continuous with a midline structure called the third ventricle. So there's the third ventricle sitting in the midline, and then the third ventricle is connected by a thin channel to the fourth ventricle. So let's draw the third ventricle, and that's connected in turn to the fourth ventricle via this thin channel. And then the fourth ventricle is continuous with the spinal cord, becoming what's known as the central canal of the spinal cord. So let's just recap those names. Let's get rid of all this. So what we said that we have got the lateral ventricle, and there are two lateral ventricles, one on each side. So there's a lateral ventricle. We have got the third ventricle, which is this one there, sitting in the midline. You've only got one third ventricle. We've got what's known as the Cerebral Aqueduct. So that's that narrow channel, that narrow channel connecting the third ventricle with the fourth ventricle. So the fourth ventricle is this one here. Okay? And cerebrospinal fluid typically flows in the caudal direction. So the CSF typically runs in this direction there, down towards the spinal cord. Now, how does the CSF flow? What you've got to realise is that the ventricular system, its walls are not muscular, so that we don't have peristalsis occurring. What actually happens is it just moves essentially by bulk flow. And it moves in that way because CSF is constantly being produced. So in all of the ventricles we have these highly vascular structures, which I'm drawing in red. And these structures look very much like bunches of grapes when you look at them in life. And if you pick one up in a living person, you see it constantly dripping CSF. And these structures I've drawn in red are known as the choroid plexus. The choroid plexus. And the choroid plexus is the structure which is constantly synthesizing CSF. As it happens, the choroid plexus of the lateral ventricle is the biggest, and so therefore the most CSF is made in the lateral ventricle. Hence why the CSF travels in this cranial to caudal direction. So we've considered how CSF is made and the direction in which it travels, but we haven't answered all of the questions which arise from this. Now, the obvious question is, if CSF is made, does it get reabsorbed, or how does it get out of the ventricular system? Because we've got a problem, haven't we? If you think about this system in detail, we've got a problem. The central canal of the spinal cord, so that caudal continuation of the fourth ventricle, the central canal of the spinal cord, is incredibly narrow. You know, it's a tiny little channel, less than 1 millimetre in diameter. And the resistance that this channel puts against the flow of CSF is incredibly high. And what that means is that CSF does not simply continue down into the spinal cord. Most of it could not flow through the central canal of the cord. So it must be getting out some way else, a different way. And in order to understand how the CSF escapes from the ventricular system, we need to do a different view. This view that we drew was a coronal section of the central nervous system. What we need to draw instead is a sagittal section. So we're going to draw a sagittal section in the midline. And by doing that, we should get to understand how CSF is able to escape from the ventricular system itself. So what I'm drawing here is the brainstem. So I've drawn the brainstem here, particularly the region around the fourth ventricle. So the region we're focusing on is this region here around the fourth ventricle. So this is the brainstem, which is the junction between the pons and the medulla. All right. This end is caudal, so that's more towards, you know, the back end of the body, the tail, as it were, and this end is cranial. Sitting dorsal to the fourth ventricle, we've got the cerebellum. So this is the cerebellum here, sitting just on the back of the brainstem there, and here is the central canal of the spinal cord, and that opens out, as you'll remember, into the fourth ventricle. All right, so here is the fourth ventricle there, and this cavity here is the cerebral aqueduct. So for all intents and purposes, the fourth ventricle is the end of the line when it comes to cerebrospinal fluid. So how is it going to escape? Well, the way it escapes from the ventricular system is that actually in the walls of the fourth ventricle there are three openings. And I'm going to draw one of them here. There's one in the midline, and then there are two lateral openings. And it is through these openings that the CSF can escape. So here comes the CSF down from the third ventricle via the cerebral aqueduct into the fourth ventricle, and then it is able to escape from the ventricular system. If we did not have these perforations in the fourth ventricle, the pressure of CSF would back up to dangerously high levels within the brain, causing the disease known as hydrocephalus. So that is why we have to have these perforations in the fourth ventricle. If we didn't have an exit route for CSF, then hydrocephalus would result. So this CSF escapes from the fourth ventricle, and of course it escapes into the subarachnoid space. So here's the arachnoid, which I'm drawing onto our sagittal section. So that CSF escapes into the subarachnoid space to then bathe the external surface of the brain and spinal cord. Now, of course, the next question to me is going to be, well, how does the CSF then escape from the subarachnoid space? And that's a topic which I'm not going to go into in detail, apart from to say that there are structures associated with the dural venous sinuses, typically sitting up around here, around the superior sagittal sinus, and these are the routes whereby CSF can escape from the subarachnoid space into the venous blood. So essentially all you need to really know is that CSF ultimately gets into the venous blood through these so-called arachnoid granulations. So these are called arachnoid granulations. That is how CSF escapes into the venous blood via the arachnoid granulations. So I hope that this simple introduction to the cerebrospinal fluid and the ventricular system was helpful. The reason I've included this is because later on in the module we're going to be thinking about brain swelling, and to understand brain swelling you need to know something about CSF and its distribution. Okay, thank you.